So let's revisit that question I asked you at the beginning. Why do you eat? Well, it should be obvious now that you have to eat so that we can digest those food particles down into their smallest components that we can use to make ATP. Or we can store those molecules so that we can use them later, whether it's for energy or it's building body structures or building hormones, right? So your liver, remember, stores lots and lots of glucose by hooking them together to make a long-chain polysaccharide called glycogen. So I often will tell students to think of glucose molecules like Legos. And if you're going to store your Legos, it's much easier to be able to get them out again and use them if you hook them all together, rather than just taking all your Legos and throwing them into the toy box and you have to go find them when you need them. Let's put them all together and make a big block of Legos. So that's sort of what your liver does with the glucose molecules. It hooks them all together to make a big structure called glycogen. So when your liver gets low on glycogen, it's time to eat. If you don't eat, your body's going to start catabolizing triglycerides, fats, and proteins. The body normally catabolizes some of its triglycerides and proteins, but large-scale triglyceride and protein catabolism doesn't happen unless you're starving or you're eating very few carbohydrates. Sounds like a fad diet there. Or if you're suffering from an endocrine disorder. So the glycerol part of triglycerides, lactic acid, and some of the amino acids can be converted in the liver to glucose. The process where glucose is formed from these non-carbohydrate sources is called gluconeogenesis. Lipids, fats, are nonpolar, so they're hydrophobic. You should have learned this before. Hydrophobic, water-fearing. So they don't dissolve in water. To be transported in our blood, which is mostly water, hydrophobic molecules have to be made more water-soluble by combining them with proteins that are produced by the liver and the intestine. So the lipid and the protein complexes that are formed when this happens are called lipoproteins. There are different types of lipoproteins and they have different functions, but they're really all just transport vehicles. They move things from one place to another. So they provide delivery and pickup services so that the lipids can be available when body cells need them, or can remove lipids from circulation when they're not needed. Lipoproteins are categorized and named mostly according to their density, which varies with the ratio of lipids, which have a very low density, to proteins, which have a high density. So the four major categories of lipoprotein, from the most fat, lowest density, to the least fat, highest density, is listed here on the slide. Let's look briefly at those four. Chylomicrons transport dietary lipids out to adipose tissue for storage. They only contain about 1 to 2% protein. So that's a whole lot of fat, right? Remember, fat has low density. So chylomicrons enter the lacteals of the intestinal villi, and they're carried in the lymph, the lymphatic fluid, to the venous blood, and then into systemic circulation. Your very low density lipoproteins, VLDLs, form in the hepatocytes, and they're mostly endogenous or made in the body lipids. They only contain about 10% of protein. They transport triglycerides that are made in the liver out to adipocytes, fat cells, for storage. LDLs, low-density lipoproteins, have about 25% protein. They carry about 75% of the total cholesterol in the blood and deliver it to cells throughout the body for use in making and repairing cell membranes and making steroid hormones and bile salts. When your LDLs are present in excessive numbers, they also deposit cholesterol in and around the smooth muscle fibers in your arteries, and that forms those fatty plaques that increase the risk of coronary artery disease. So it's because of this that the cholesterol in LDLs, called LDL cholesterol, is known as the bad cholesterol. So if you eat a diet that's high in fat, it can increase the production of VLDLs, which can in turn elevate the LDLs and increase the formation of fatty plaque. Your HDLs, high density lipoprotein, remember more protein means more dense, your HDLs are 40 to 45% protein, so they help to remove excess cholesterol from your body, from the blood, and transport it to the liver so it can be eliminated. Most of the HDLs prevent accumulation of cholesterol in the blood, so a high LDL level is associated with a decreased coronary artery disease risk. So it's for this reason that the HDLs are known as the good cholesterol, or as I usually teach, L, lousy, H, healthy. There are two major sources of cholesterol in the human body. Some of it's present in the food that we eat. Dairy products, meats, beef, pork, processed lunch meats, eggs, all the good stuff, right? But most of it is synthesized by the hepatocytes in the liver. In the United States, blood cholesterol is usually measured in milligrams per deciliter. A deciliter is 0.1 liters or 100 milliliters. For adults, desirable levels of blood cholesterol are what we call total cholesterol, under 200 milligrams per deciliter, with LDLs to be under 130, HDLs over 40, remember H is for healthy, we want more of those, but your triglycerides can range from 10 to 190. So as total cholesterol level increases, the risk of coronary artery disease also increases. When your total cholesterol is over 200 milligrams per deciliter, the risk of a heart attack doubles with every 50 milligrams per deciliter of increase in total cholesterol. Total cholesterol of 200 to 239 milligrams per deciliter with LDLs of 130 to 159 are borderline high. Total cholesterol that's over 239 with LDLs over 159 are classified as high blood cholesterol. Now, if you're in my class, I don't necessarily want you to know those numbers, but I want you to realize that HDL, it should be in a healthy cholesterol level. Your HDL, you want it to be higher. You want your LDL to be a lower number, and also anything over 200 puts you at higher risk, especially if you have an increased risk of LDLs versus your HDLs. So there are several types of therapy that can be used to reduce blood cholesterol, and that includes diet and exercise, and some pharmaceutical drugs. Regular physical activity, especially aerobic level activities, is going to raise your HDL. Dietary changes try to reduce the total intake of fat, saturated fats, total fat, cholesterol. Drugs that are taken to treat high cholesterol promote the excretion of biles in the feces, some of them, and others are the statin drugs, which block the enzyme that's needed for making cholesterol.